You know, when you bite into an apple or plant a seed in your garden, do you ever stop and think about the incredible story behind it? It's not just a simple biological process. It's this high-stakes drama of survival, incredible journeys, and amazing transformations. So let's dive into this fascinating world of sexual reproduction in flowering plants.
I think for most of us, flowers are just, pretty things to look at. But this quote, it totally flips that idea on its head. To a biologist, they're not just decorations. They are masterpieces of engineering, designed for one of the most important jobs in nature. They are the stage where this whole drama of creating the next generation gets to play out.
So what is that true purpose? Well, every single part of a flower, every petal, every color, every scent, it's all part of this super sophisticated strategy for one single goal: sexual reproduction. The entire structure is built for this mission. So let's meet the key players in this story.
All right, so in this story, you've got two main characters. On the male side, you have what's called the andrecium, which is basically all of the stamens bundled together. And on the female side, you have the gynecium, which is made up of one or more pistils. These two structures are where all the action begins.
Okay, let's get a closer look at the male part, the stamen. It's got two parts, this long, skinny stalk called the filament, and then this really crucial part on top, the anther. You can think of the anther as the factory. This is where our little traveler, the pollen grain, gets produced by the millions.
And this is the destination. The female part, the pistil, is perfectly designed to receive that pollen. Up top, you've got the stigma, which is like a sticky landing pad. Then there's the style, a tube that connects it all. And down at the base is the ovary, a protected little chamber that holds the really precious cargo, the ovules. The entire goal of this whole process is to get that pollen from the anther to this stigma.
But before this epic journey can even start, our traveler, the pollen grain, has to be created. So how exactly is it made? Let's go back inside that anther factory and see how it's born.
The whole process is called microsporogenesis, which sounds complicated, but it's actually pretty elegant. It starts with a bunch of cells called sporogenous tissue. Through a special type of cell division called meiosis, these cells form little four-packs called microspore tetrads. Then, as the anther matures, these tetrads break apart and each one grows into a tough, independent pollen grain, all suited up and ready for its mission.
You know, it's so easy to think of pollen as just dust, but it is so much more than that. It's basically a tiny, self-contained delivery vehicle for genes, and it's built like a tiny fortress. I mean, check out its two-layered wall. That outer layer, the exine, is made of something called sporopollenin. This stuff is one of the toughest organic materials known to science. It protects the vital cells inside from pretty much everything: heat, acid, you name it. It's a biological suit of armor for a very important journey.
And this shows us something really interesting about when our pollen grain actually starts its trip. In over 60% of flowering plants, the pollen leaves home, so to speak, in a simple two-celled state. But for the other 40%, it's a little more prepared. That generative cell is already divided, so it starts its journey in this three-celled, ready-to-go state.
So the pollen is made, it's armored up, and it's ready to go. Now begins the most dangerous part of its life, the journey. This is pollination, getting the pollen from the anther to the stigma. And trust me, it is fraught with challenges.
Plants have evolved three main strategies for this. Autogamy and Geitonogamy are basically forms of self-pollination, but the real game changer, the one that really matters for evolution, is Xenogamy, moving pollen between completely different plants. This is the only one that introduces new genetic variation. And why does that matter? Well, that genetic shuffle is the key to resilience. It gives a species the tools to fight off new diseases, adapt to a changing climate, and ultimately to survive in the long run.
So how does the pollen actually travel? It can't move on its own, it needs a ride. Sometimes that ride is something non-living or abiotic, like the wind, which is exactly why grasses have small, unremarkable flowers. They don't need to be flashy. Or it can be biotic, using animals. And to attract them, these plants evolved large, colorful, fragrant flowers that produce nectar. It's basically a reward for the delivery service.
But here's one of the biggest dangers of all. The clock is ticking. For some of our most important food crops, like rice and wheat, that pollen grain is only viable for about 30 minutes after it's released. That's it, 30 minutes. It's an incredible, frantic race against time to find the right stigma before it's too late.
So, our little pollen grain has survived the journey, it's landed on the right stigma, and it's grown a tube all the way down to the ovary. Now we've reached the climax of our story, a truly remarkable event that is found only in flowering plants.
The pollen tube, guided by these tiny chemical signals, finds its way right into the ovule and enters something called the embryo sac. It then breaks open, releasing its precious cargo, 2 male gametes. And this is what sets the stage for a biological event so unique it gets its own special name.
This event is called double fertilization, and it's double for a very specific reason. Unlike almost anywhere else in the living world, flowering plants carry out 2 separate fusion events at pretty much the exact same time. Let's take a closer look at what happens.
Okay, so think of it like a 2-act play happening in a split second. First, one male gamete fuses with the egg cell. That's Xenogamy, and it creates the diploid zygote, which is going to grow into the plant embryo. Then, the second male gamete fuses with 2 other nuclei, and this creates something truly unique, the triploid endosperm. That triploid nature makes it an incredibly rich and efficient food source, like a supercharged lunchbox for the embryo. This little trick might be one of the secret weapons that allowed flowering plants to so successfully take over the planet.
This incredible double fertilization event triggers a whole cascade of changes. The mission is complete, and now the flower begins its final transformation, creating the legacy that will carry its genes on into the future.
And you can see that transformation perfectly here. The zygote becomes the embryo, the primary endosperm cell becomes the food supply, and on a bigger scale, the entire ovule develops into a seed, while the ovary that was protecting it swells and ripens to become the fruit.
So here's a fun botanical fact. A true fruit, botanically speaking, like a mango, develops only from the ovary. But sometimes, other parts of the flower get in on the action. In an apple or a strawberry, the fleshy part we eat actually develops from a part called the thalamus, so we call those false fruits. But whether they're true or false, the fruit has a brilliant ulterior motive. Get an animal to eat it and carry the indigestible seeds far, far away. It's a genius strategy for dispersal. And the legacy of this process, the seed, is built to last. How long? Well, the oldest viable seed ever found was from a lupine, excavated from the Arctic tundra. It successfully germinated and flowered after an estimated 10,000 years of being dormant. Just think about that, an absolutely incredible feat of biological preservation.
Now, of course, nature loves to have its exceptions. Some plants have found a shortcut. Parthenocarpic fruits, like the banana, develop without any fertilization happening at all. This is exactly why the bananas you buy in the store are seedless. They bypass the entire process we've just talked about.
And there's an even stranger shortcut called Apomixis. Here, plants like dandelions actually produce seeds, but they do it without any fertilization. It's a form of asexual reproduction that cleverly mimics the outcome of sexual reproduction. It's a fantastic strategy for a plant that's found a perfect, stable place to live. It can just rapidly produce clones to colonize the area.
And this brings us to a final, really profound thought. Think about agriculture. We create these amazing hybrid crops, but we have to recreate them every single year because their seeds don't grow true. But what if we could transfer the gene for Apomixis into these hybrids? Could we essentially lock in that perfection, creating seeds that are perfect clones of their superior parents? It's a question that connects this amazing natural story directly to the future of our food. Could this asexual mimicry be the key to feeding the world?